Hi, I'm Ahmad Afiq Izzuddin bin Sama Jamal, and I will be voicing the materials needed and the experiment procedures.
My name is Sharifah Nurzaida Izzati and I'll be doing the experiment and also the observation part.
Hi there, my name is Kok Sang Yi and I'll be talking about title, learning outcomes, and introduction. So let's start. Hello everyone, we are from Group 10 Cooler 8 and today we are going to talk about DNA extraction. There are two learning outcomes for this DNA extraction experiment. The first one is to understand the principle of DNA extraction. After completion of this experiment, we can know the function of the chemicals used in this experiment. We can know how the chemicals play their role in the procedure, as they all have their unique properties that allow them to be part of the experiment. We can know why the chemicals are kept cool in this experiment and the correct way to spool the DNA so the DNA strand will remain intact. Next, we can understand the procedure of DNA extraction. We can know the exact procedure to complete the experiment for a better result. We can also apply the precaution steps in the experiment and keep an eye on them so the result will be more accurate. Now we're going to look at the introduction of the experiment. So what is DNA? DNA is known as deoxyribonucleic acid and it is the genetic material for all living organisms. So every organism consists of many cells, and in the cells there is presence of nucleus, and in the nucleus there is DNA. DNA encodes for many aspects of the organisms such as development, growth, and structure, and it even encodes for the protein in the human body. DNA extraction is the technique used to isolate DNA in the biological sample. So it uses a combination of both physical and chemical methods. This is pivotal in many fields, such as biotechnology and forensic analysis, where the structure of the DNA has to be known for all the scientists. So when the scientists can better understand the structure of DNA after they've extracted the DNA, they can make many improvements to the human life, such as in the medical sector, to cure many diseases, such as those that are hereditary.
The materials for these experiments are a plastic bag with a back lid, a half glass of water, a jar, a bowl, some strawberries, some dish soap, a sieve, some salts, tablespoons and teaspoons, and a rubbing alcohol. First, add half a teaspoon of salt into a plate, and then a tablespoon of dish soap was added to the salt. And then half a glass of water was added to the mixture and was stirred well. Some strawberries are put into a plastic bag. A few tablespoons of the mixture was added into the plastic bag. Next, the air in the plastic bag will be pushed out, and the plastic bag is then sealed. The strawberries are then mushed until it became a fine and foamy paste. Then the strawberry pulps were strained into a jar using a sieve. the same amount as the strawberry juice, and that's it for a while.
After the alcohol is added into the mixture, we can see that there is a separation between the alcohol and also the mixture, and then the DNA start to form on top of the mixture. So as you can see, the snot-like thing, which is clear in color, that is the DNA of the strawberry.
Hi, my name is Nur Alfa Shakira binti Muhammad Suharto, and I'm going to present results and question experiment 3A. So next are the result and questions. Firstly, state the function of each chemical or components below in DNA extraction. For salt, the function is to extract the DNA. For glycogen, it will mix the cell to form open, therefore DNA will be released to the solution. And for the ice-cooled alcohol, it can facilitate in precipitation of DNA to occur more quickly. Why is the onion mixture processed through the blender? Because it can increase the total surface area so the onion cell has more surface exposure. And the second one, why do we need to cool the mixture in ice water bath? Because it can avoid the DNA cell from being destroyed. And for the conclusion, from the experiment of extracting DNA from strawberry, the combination of physical and chemical methods can be conducted in purification of DNA. The choice of using strawberry is because it has 8 copies of chromosome, that means it has a lot of DNA to isolate. This proves that DNA can be extracted from any kind of cell. So this is the reference that we got from our research on our experiment. That is all for me.